My talk, as Chris said, is about setting up a bioinformatics QC pipeline. And let's just get to it. So I'm the bioinformatics specialist for the SBC here at UBC. And we're part of the VPRI, which is the Office of the Vice President, Research and Innovation. And so I'll start with this slide where there seems to be a reproducibility crisis in science. And so this is kind of something that has come up in, it's been a thing for a long time, but it's something that's been talked about in more recent years. And I'll just read out the quote here. So more than 70% of researchers have tried and failed to reproduce another scientist's experiments, and more than half have failed to reproduce their own experiments. And I'm sure if you've worked in the lab or if you do any type of informatics analysis, you can tell if you try to reproduce people's analysis, it's not always super straightforward. And this is one of the main reasons why a pipeline is necessary, especially with certain processing is very, very involved and has many steps. And so it's very important to have a reproducible kind of pipeline setup. And so this is just another slide to explain that it's not just biology. It's chemistry, it's physics, it's medicine, earth and environment, and other faculties that experiment where they experience experiment failure and lack of the ability to reproduce the experiments of others. And because of this lack of reproducibility, there is also some difficulties in publishing. And so yeah, so I'll start with the background. I can give you guys a kind of an overview of who I am. And so my formal education is from SFU in the molecular biology and biochemistry stream. And so I actually had worked, after graduating from SFU, I had worked at a place called the Vancouver Prostate Center for about 7 years as a research technician initially, doing lab work. And then after that, I moved more so into the computer side of things and started helping with data processing and building pipelines over there. And then more recently, about a year ago, I joined UBC over here at the SBC. And the first thing that I was tasked to do was to build a bioinformatics QC pipeline. And so I'm going to kind of go through what that looks like, what goes into the thought process of when you're creating one, like roughly how to write one, and then what kind of metrics we're currently tracking with regards to QC. And then really briefly the implementation and conclusions from that. So what is bioinformatics? It's kind of the intersection of a whole bunch of different things. So it's computer science, it's chemistry, biochemistry, biology, statistics, math, engineering. So there's a lot of overlap. And so I kind of view this as the best bioinformatician would be essentially a unicorn, so they don't exist. There's no such person that has an in-depth knowledge of each one of these things, but there is definitely a need for knowledge in computer science, biochemistry, biology, and statistics. And so that's kind of an overview of what bioinformatics is. And so specifically what we deal with and what I personally deal with on a daily basis is what's called next generation sequencing, both DNA and RNA type sequencing data. And I just have the workflow, which is really oversimplified here, but essentially you start with a sample input, and that goes through lab-side QC. And then there's a sample preparation, which converts the material, whatever it may be, if it's DNA or RNA, various nucleic acids and different forms of them. It goes through some form of sample preparation. And then after the sample preparation is done, there's more QC that's done still in the lab. In this case, it's called library QC. And once that is all passed, then you then load the libraries onto the sequencer. And we deal mainly with Illumina type sequencers, so MiSeq, NextSeq, or HiSeq. And we're looking at getting into the NovaSeq for much higher throughput type data. And then finally, that produces data, which is my favorite part because that's what I do. And so there you go. And so what exactly is next-gen sequencing data? So it's a high throughput technology. What that means is it generates millions of reads, and these reads are just strings of G's, A's, T's, and C's. And they have associated quality values in the case of Illumina data. And so this is exactly what a short read would look like. So you have The first line is just information about the sequencer and location of that fragment of DNA on the sequencer. And then the second line is the actual sequence of data. The third line is just a delimiter, it's a plus sign, and then the fourth is the associated quality values with those bases. And in this case, this is just a 5x control DNA sequence. And so you can imagine it'd be difficult if you had to do this by hand or if you had to manually reproduce someone's analysis because you have a million or millions of these reads. So it'd be best to kind of work that into a reproducible pipeline of some sort. And so this is just another slide to explain that it's only getting more and more necessary to have this kind of setup for any data processing, specifically but for bioinformatics. And in this case, it's showing just the increase in output and the decrease in price. So since it's getting cheaper and cheaper every year and the sequencers are getting higher and higher throughput, it just becomes more of a necessity to be able to control these processes. And so when I was first tasked with figuring out writing this QC pipeline, there was the problem of looking at different pipelining tools. And so there are many out there. And here's kind of what they are and why we want to use them. So they're workflow management systems, and they have to be written in a way that's reproducible and scalable for data analysis. And a lot of the times they work on the basis of rules, which are essentially structures to allow inputs to be fed in, and then some processing to happen, and then that yields some output. And then that kind of goes along in a chain, which I'll show later some real examples as well of what that looks like. And so we had to assess what were the needs of the SPC and specifically for pipelining. And so like I said on the previous slide, it had to be reproducible, it had to be scalable, it had to be efficient, so preferentially parallelizable, portable, so on different various systems, both on cluster and single node systems, and obviously automated would be the best. And so our wants were ease of development. So in my case, I have my strength in coding is in Python as well as Bash. So that would be kind of preferable, as well as Unix compatible, because most of the time the bioinformatics tools are written for Unix systems. And obviously free, because everybody loves free. And so I compared these three tools after talking with some people in the community, and so these were the three that I had looked at, and Galaxy was not really amenable because it was more so, like the graphical user interface version of it especially wasn't something I wanted to do. I wanted to be more scalable, and the GUI version of it wasn't just going to cut it for us. And the Rufus was a Python-based pipeline manager, but it just didn't seem as intuitive as the final one here, Snakemake, which is the one that I ended up choosing in the end. And so with Snakemake, I'm going to go through kind of some examples of some toy examples of how you would write specific rules and what that entails. So the resources that I had available to me, which I think a lot of you have access to as well, is a Compute Canada resource. Specifically, in my case, I was developing in Cedar, which is based over in SFU, not too far from UBC here. And it would, it's a beast of a machine. So you have access to a max of 58,000 cores, 300 terabytes of RAM, and each user gets 10 terabytes of scratch space. So that's high performance, high speed disk space. So yeah, that was definitely more than enough to do this development as well as running the pipeline on a regular basis. And so the people that helped me do this were the two key people from the Advanced Research Computing team, Jamie Rosner, who used to be a bioinformatician, and Venkat Mahadevan, who is a sysadmin over there, got me access to this resource, as well as Dr. Helen Burt from the VPRI. And so Snakemake specifically, it decomposes workflows into rules. And I'll go into greater detail in a few slides. These rules define how to obtain output files from input files, and the Snakemake infers dependencies and execution order. So once you've built the pipeline, you can restart it from any stage. So if you process to halfway point, you can actually start from that halfway point and continue on with this software. And also, I'd like to mention that Snakemake is free, freely available, and it's open source. And so I like reading comics on XKCD, but this is a good one to show kind of where Snakemake falls in this little graph. So tools that need a manual is this middle point kind of thing, and then tools that need a manual but don't have one, and tools whose manuals start with how to read this manual, those create problems. And so I found that Snakemake was fairly easy to develop in once you kind of got over a certain hill of understanding how it's supposed to work. And so it was able to enable me to solve problems more than create. And so now for something completely technical. So it's very simple. I mean, the main structure of how Snakemake works is extremely simple. So you have a rule, which you have a directive here as a rule, and then you give it a name. In this case, the name's sort. And then you give it some input and some output, and then you can either run the shell or Python or various different ways of interacting with your environment. But in this case, so you have an input that's dataset.text, you have an output that's dataset.sorted.text, and then here you can call your input and output using these curly braces. And so it's just sorting the input and piping it to the output. So very simple to write in Snakemake, which is one of the best parts of it. And so you can also generalize these rules with named wildcards. So you can make it a little bit more complex by adding curly braces to the input and output here. So it'll essentially, Snakemake will automatically look in the directory path2 and find anything that has .text, and will run this rule on all files that have the beginning of .text and output with the same prefix. It'll be the prefix.sorted.text. So it's very easy to make it amenable to sets of data, which is obviously what you want. And so you can also specify multiple inputs and outputs. And so in this case, it's sort and annotate is the rule name. The input there are two, which it's comma separated. And so you have one that has a wildcard, one that doesn't, and then the output is just dataset.sorted.text. And so in this case, you're sorting the input 0. So in this case, you can call it by position. So index is the first one. So it's Python, and Python is 0-based indexing. So this is 0, and this is 1 for the index. And so you sort input 0 and input 1, and pipe that to the output and paste those together. And that creates this output here. And so you can also do it by name instead of by index. And so you see here that the input has names A and B, and so you can then call them using a separator. So input.A will call path2 dataset.text, and then input.B will actually call this second line here. And it's the same rule, but it's just slightly different way of creating it. And so you can also run Python within these rules. So instead of shell directive down here, now it's got run. So essentially the same inputs and outputs as the previous rules, but you can run Python. In this case, it's just a with open statement. And so here's an example of a real rule that's actually implemented in our DNA QC pipeline. And so this is just a step that's mapping DNA reads to the genome. And so one thing I didn't mention in the previous slides is that you can actually use functions for the input and the parameters. And this is getting quite a bit more complex than the previous examples. But in this case, you have an input, which is get trimmed read. So in my case, I was wanting to have it automatically detect whether it was single end or paired end. And in this case, it was also looking for trimmed reads. And then you can also have this temporary directive here, which actually, once this rule has been run, will then remove or delete this file from your system that you're interacting with. And then you also have a logs directive and a parameters directive. And so the logs, you can then pipe any standard error, which is what this to greater than to log. That's what that means down there. And so in this case, I'm using inputs, outputs, logs, parameters, And in the case of the parameters, I'm also using functions. And so in this case, since I wanted it to be dynamic, I have different genomes that I'm mapping to. Get genome index actually figures out from a configuration file that I give with sample data set what genome it's trying to map to. And then the RG is just the read group, if you're familiar with that. Finally, there's also a threads directive, which is essentially how many CPUs you want to use for this rule. So some bioinformatics tools are multi-threaded. In this case, BWA, which is the mapping tool, is multi-threaded. And so you can actually use this -t directive to make it threaded. In this case, I'm using 46 cores to map this, which is quite a bit. And then, yeah, so I guess to go through how job execution works, for Snakemake, job execution works only if the output file is the target requested and does not exist. And so what that means is if you have input files and the output files don't exist based on a rule that's part of your Snakemake pipeline, it will create those outputs running it through that rule. Number 2 is output file needed by another executed job. So if there's a downstream job that doesn't have its output, then it will run the upstream job to create that output. Number 3 is the input file is newer than the output file. So if you update your data set for your input side, then it will automatically detect that it wants to rerun your pipeline, so it's using the newest data possible. And number 4 is the input file will be updated by other jobs. So if there's some process that touches a file and updates it in some way, then that will then be rerun through the pipeline after running it again. And the final way is, of course, if you force execution. So if you just wanted to rerun something, even though the first 4 rules weren't actually true, that's the final way you can do it. And then also there's a cluster execution. So the way I've written it is using pipeline profiles. And so that essentially allows you to submit jobs to the cluster. If you're familiar with that kind of environment, so it's just a high-performance computing environment. Cedar is one of them. Essentially, you have a head node that you log into, and then from there you can launch jobs to the compute nodes. And so Snakemake allows that, so you can actually use this for your pipelines to actually process your rules, and each rule can be sent to its separate cluster, or sorry, compute node on the cluster. And so this is by way of executing a DAG, which is a directed acyclical graph, which I'll show an example of in the next few slides. And like I said, it's by cluster job submission. And you can configure this as granular or as general as you'd like. So you can set max jobs at a time that can be launched. You can set CPU max, mem max, and like I said, it can be general or granular. So you can set that, you know, for the entire pipeline. You can set CPU mem and max and mins. You can also set that per rule. So you can actually set, if you have one rule that's really CPU intensive, you can increase the max just for that rule if necessary. So you can control the amount of resources you're requesting, which is great. And so to give an example of the metrics that we're tracking here for QC in terms of bioinformatics in our lab, here's just a list of them. So things like adapter trimming, the duplicate rate, percent aligned for genomes we can map to, insert size, coverage, error rate, GC content, and that's mainly for all types of data, mainly DNA, and then also for RNA specifically, we're looking at strand specificity for the strand-specific libraries, and 5' to 3' bias, and percent rRNA, intron-exon ratio. So, you know, there's a lot of things being tracked, and so in order to do this in a systematic way, it's kind of one of the reasons why a pipeline is necessary. Because the pipeline also allows you to know what versions of the tools you're running and what parameters are set, which is just really important if you want to be able to reproduce certain analysis. And so this implementation, it's done in a conda environment here at the SBC, which works well with Snakemake as they're both Python type tools. And it's also version controlled using GitHub. It's not publicly shared but it is version controlled. So every time there's an update to it, we can go back if something fails, or we can track what kind of updates we've done if we've updated software, and then we can see what the differences are. We can run test data sets through each version to see that there's not any differences, or if the differences are there, that we expect them, and then we know what they're supposed to be. And so right now, the SBC has 3 QC pipelines. It's combined into one that's dynamically determined, so it doesn't really matter what I give it as input. It will automatically detect if it's paired or single end, and if it's RNA or DNA. This is based on a configuration file that's fed to the Snakemake pipeline. And so here's just a simple example of 2 samples going through a DNA QC workflow. And so the initial inputs are kind of from the top down, except for these fast QC steps. So each one of these represents a rule. And so this is an example of a DAG, so a directed acyclic graph. And so it's acyclic because it's going in one direction. And so here you can see I'm doing some trimming on a sample. And then once this is trimmed, I'm doing some screening for contamination and cross-contamination, as well as other species being detected using FastQScreen. And then I'm piping this trimmed data through to the mapping step using BWA-MEM. And then from there, I'm doing some duplicate marking using Picard. And then once I've got the duplicates marked, I'm then passing that along to some BAM-QC, as well as some GC bias and WGS metrics in Picard, and also FastQC for the raw data here. And all this gets piped into multi-QC, which is just a great tool to visualize any type of QC information in the bioinformatics world. It essentially outputs a HTML file, which is very visual and interactive. So it really gives you a good sense of how your data is performing. And I highly recommend using that for any type of QC and bioinformatics. It's a great way to compile a lot of information into one place. And then here I have a couple different things, because I copy back some of the information from multi-QC that it parses out, and then do some final cleanup of the files, and then that parses to all. And so that just allows it to call all the upstream steps. And so, like I said, this is kind of a relatively simple pipeline, even though there are quite a few steps per sample. Here's an example of one that's not as simple. So I think I counted there's something like 28 steps for this pipeline. And I think this was a SNP calling pipeline with recalibration steps. So, you know, this is just another reason why you want to make this as reproducible as possible, because if you had to do this manually, you'd have to take very good notes because each step has its own parameters and its inputs and outputs and log files that are outputted. And so being able to track that by hand or manually is extremely difficult. So using some pipelining software is actually very useful in this case. And so, yeah, that's pretty much all I want to say for that slide. And finally, some conclusions. Apologies for this being quite a short talk. But so Snakemake satisfied all the needs of the SBC and is simple to work with. The complex metrics the SBC is most interested in are being tracked in an automated fashion. And also the implementation allows for reproducible, scalable, and flexible and trackable QC. So we're now using the outputs of all our runs that we're processing to monitor quality on a run-by-run basis, but across time as well. So we're able to detect outliers quite easily using this kind of tracking and pipelining. And so, yeah. There's also, I had done a workshop back in the summer, so here's a link. So it's kind of an interactive workshop that kind of goes more into depth on how to set up a Snakemake pipeline using some actual data and some rules, and it goes kind of step by step how to build. I think it was a SNP calling pipeline that I had gone through, very rudimentary, not one that I would actually implement, but it was more so for just as examples on how to build Snakemake pipelines. So I'd recommend checking out this bit.ly link. It's just a link to my GitHub there. And yeah, that's actually all I had to say for today.
Wow, that's great, Brian. You know, as someone who used to use Make a lot, make. The GNU Make is definitely one of those softwares that needs a how to read this manual before being able to use the manual to use the software. So I appreciate how Snakemake, it's very, very readable. It follows, there seems to be movement in that direction with using formats like YAML, where you get this extremely readable structure for understanding what's going on in a process, and so that was really cool. I'm going to open the floor to some questions. Feel free to unmute, but I'll ask my own question first. Python's known for being picky about indentation, so I assume that Snakemake is also very picky about that too. Is that correct?
Yeah, it's written in Python, so it's equally picky about indentation for sure. Yeah.
Yeah, and the pipeline visualizations were those were taken from publications, or is there a tool that will help visualize some of those where you set up your pipeline and you get that sort of flow chart, or that view of your DAG, your graph? Is there tools to visualize those in Snakemake?
So Snakemake has a built-in way to build those. It's I think it's just a dot type format that it then converts to SVG.
Okay.
And so it actually, based on your rules and your data input, it actually outputs those automatically if you request it.
Yeah, very cool. So are there any other questions out there?
Yeah. So Brian, I have a couple of questions. So first, would you see it as installed as Snakemake software on a cluster, and then you use it from your Python environment, or it needs to be a part of your Python environment installed as using Conda?
I mean, you could do it either way, but the way I have it is I have it installed through Conda, so I have the control over the version and everything. And if I want to update, it's fairly easy, so I have it in my own Conda environment on the cluster.
Right. And second question is, apparently you can write a pipeline as a Python script calling all the steps required. So what would be the most beneficial difference between Snakemake and just writing it as a Python script?
So there's a lot of behind-the-scenes things that Snakemake is taking care of. So like I was saying, let's say you have a data set, you have 10 samples, you run your normal script, then you want to run another set of 10 samples, but only to a certain step. You can actually get Snakemake to get the specific samples. You can call specific samples to get to a certain step, which might be more difficult depending on how you write your pipeline. And it's just more flexible, I find, and easily scalable. So yeah, I mean, one of the biggest things was just handling all the directory structure you can set up very easily with Snakemake, and then also just getting it to reprocess. So if you wanted to reprocess to, I wanted just to get BAM files or something, you can just specify an output and it'll just run what you need to get to that output. And there's no extra writing you need to do. It's just a simple command.
So Chris, can I have another question?
Yeah.
So Brian, in practice, because you didn't show this, how do you use the Snakemake scripts? Do you call Snakemake and then the script name, or what is the pipeline?
Yeah, so there are a couple different ways you can call it. The way I have it set up is using the cluster profiles. So in that case, you just type snakemake -- profile and then the name of your profile, which is set up in config location, and that specifies all the rules and all the dependencies, all the tools and everything like that. You can also set it up so it's run locally to the directory that you're in which case you'd have what's called a snake file, which would specify the same things, but it just wouldn't be calling it on a cluster basis. It could be calling it on a single node basis, depending on how you write it.
But the data files, how does he find the data files? They are relative to the directory you are calling it in?
Yeah. Yes, exactly.
Okay.
But you can set that up how you want. I mean, but it's usually it's a relative way. So it starts from a certain parent directory, and then it'll build it out from there.
So have you used it for non-op data?
I have not yet, but it's totally agnostic to type of, like, you don't even need to do bioinformatics with it. It's just a pipelining software, so you can actually put whatever type of data you want through it because it's just running other tools for you. It's kind of just like a wrapper for it to run more easily and reproducibly.
But do we need, like, for the nanopore, like, to to connect, like, the tools to the Snakemake, or like to download them into the Snakemake, to the pipeline?
Yeah, so you can write your own pipelines. There are some pre-made ones out there that you can check online. I can't remember what the website was exactly, but I'm not sure if anybody has any for Nanopore yet, but I'm sure someone has written some somewhere. But it should be definitely able to be written easily.
Okay. Brian, so, but you talk about reproducibility. So reproducibility comes from the profiles, right?
Mm-hmm.
So is it true? So you tell your colleagues that, well, this is the profile they should use to submit their pipeline, and that's how it is done?
Yeah. So in this case, since we're a core facility and we're processing data from many different people, different sources, collaborators, it's crucial for us to have, like, just to know what versions of tools we're running. So since I have it attached to GitHub as well, I can go back to different versions, so I have them all versions. So that allows reproducibility. So if somebody says, hey, I got data from you November 2019, like, and you have this output, how was that created? I can go back to that version of the Snakemake pipeline and tell them exactly what was used.
So GitHub is a part of that reproducibility idea. Yeah. Okay. Thank you.
Yes. Any other questions?
Okay. If there aren't any more questions, I'd like to thank Brian again for speaking today and giving us a taste of Snakemake, which I think is a pretty awesome tool for creating these pipelines. So, yes, thanks again, Brian.
Okay. Thanks, guys.
Cheers.
Thank you.